Welcome to an overview of the regulatory framework and FDA's guidance for the development and approval of biosimilar and interchangeable products in the U.S. My name is Monique Dyson, Education Program Manager at the American Gastroenterological Association. This program is a collaboration of the AGA and the Crohn's and Colitis Foundation and is part of an educational series on clinical and regulatory considerations of biosimilars in gastroenterology. This program is supported by independent educational grants from Boehringer Ingelheim Pharmaceuticals Incorporated and Pfizer. I will now introduce our speaker, Dr. Sue Lim. Dr. Sue Lim is the Director of the Scientific Review Staff with the Therapeutic Biologics and Biosimilars staff at FDA. In this role, she provides review and oversight of therapeutic biologics and biosimilar products, as well as regulatory policy development for biosimilars. Dr. Lim was previously a medical officer with the FDA's Division of Anti-Infective Products. Prior to the FDA, Dr. Lim served as an infectious diseases physician and associate hospital epidemiologist at the University Health Network in Toronto, Canada. She received her MD from the University of Toronto, where she also completed her residency in internal medicine and subspecialty training in infectious diseases and served as a lecturer in the Faculty of Medicine. She has a Master of Science degree in Health Policy, Management, and Evaluation from the University of Toronto. Thank you for joining us, Dr. Lim.
Thank you. Today we are going to be talking a little bit about the development of biosimilars in the U.S. And I've titled my talk, Overview of the Regulatory Framework and FDA's Guidance for the Development and Approval of Biosimilar and Interchangeable Products in the U.S. Here's an overview of what we'll cover over the next half hour or so. We'll start with some background and focus on the regulatory and legal background about development of biosimilar and interchangeable products in the U.S. We'll cover some general terminology to make sure everyone's on the same page in terms of what we're talking about today. And we'll also cover some general requirements for marketing applications or 351 applications for biosimilar and interchangeable products in the U.S. The next section of my presentation will focus on the development of biosimilars in the U.S., the status of where we are, and cover some specific scientific development concepts. Then we'll spend a little bit of time talking about how applicants can support a demonstration of interchangeability. And lastly, finish off discussing how to use biosimilar and interchangeable products. On March 23, 2010, President Obama signed into law the Affordable Care Act, which gave FDA the authority and responsibility to regulate biosimilar biological products, which were at that time a newly defined class of therapeutic products. The pathway to licensure for a biosimilar is described in the Biologics Price Competition and Innovation Act of 2009, or the BPCI Act. And what the BPCI Act did was create an abbreviated licensure pathway for biological products shown to be biosimilar to or interchangeable with an FDA-licensed reference product. The Act states that a biological product that is demonstrated to be highly similar to an FDA-licensed biological product, known as the reference product, may rely on licensure on, among other things, publicly available information regarding FDA's previous determination that the reference product is safe, pure, and potent, or safe and effective. And we'll cover a little bit of these definitions over the next few slides. Basically, what this means is that a biosimilar product can be licensed by FDA on less than a full complement of product-specific preclinical and clinical data, and that is what is meant by an abbreviated licensure pathway. So what is this abbreviated licensure pathway? I can tell you at the beginning what it isn't. The abbreviated licensure pathway does not mean that a lower approval standard is applied to biosimilar or interchangeable products than to originator biological products. It is, as I mentioned previously, based on the ability to rely on FDA's previous finding regarding the reference product. And this is based on a scientific determination that biosimilarity has been demonstrated. And this supports approval of the biosimilar product and allows for a potentially shorter and potentially less costly drug development program. And that is what is meant when we say that this is an abbreviated licensure pathway for biosimilar and or interchangeable products. In fact, the data package that's required for approval of a biosimilar or an interchangeable product is actually very extensive. Biosimilar applicants must submit data from analytical, non-clinical, and clinical studies to support a demonstration of biosimilarity with the reference product. And once a biosimilar or interchangeable product has been approved by the FDA, patients and healthcare providers will be able to rely upon the safety and effectiveness of an FDA-approved biosimilar or interchangeable product just as they would for the reference product that the biosimilar was compared to. So what does biosimilarity mean? It means that the biological product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components, and that there are no clinically meaningful differences between the biological product and the reference product in terms of the safety, purity, and potency of the product. Please note that both of these standards must be met for FDA to license a product as a biosimilar. What is a reference product? The reference product is the single biological product licensed under Section 351A of the Public Health Service Act, against which a biological product is evaluated in an application submitted under Section 351K of the Public Health Service Act. So let me break this down because I know there's a lot of regulatory terms here. An application that is developed under Section 351A of the PHS Act is known as a standalone application. And a standalone application must contain all information and data necessary to demonstrate that that product is safe, pure, and potent, or safe and effective. In contrast, an application submitted under Section 351K of the Public Health Service Act, this is the biosimilar, doesn't need to demonstrate that it's safe and effective. In fact, the biosimilar applicant needs to demonstrate that the proposed product is biosimilar to the reference product. And that's a different objective than the objective of a product being developed under Section 351A. So for licensure, a proposed biosimilar relies on, among other things, comparative data with the reference product, showing that it is biosimilar to the reference product. In addition, there's also publicly available information that the biosimilar applicant can rely on regarding FDA's previous determination that the reference product is safe, pure, and potent. The BPCI Act also describes what a 351K application or marketing application for a biosimilar must include in terms of data. There must be information demonstrating that the biological product is biosimilar to a reference product, that it utilizes the same mechanism or mechanisms of action for the proposed conditions of use, or indications is the most easiest way to think of conditions of use. But this is really only to the extent that the mechanisms are known for the reference product. The conditions of use, or indications, populations, proposed in biosimilar labeling have been previously approved for the reference product. So a biosimilar product cannot be approved for novel conditions of use or indications that are not carried on the reference product labeling. The biosimilar must have the same route of administration, dosage form, and strength as the reference product, and the product must be manufactured, processed, packed, or held in a facility that meets standards designed to assure that the biological product continues to be safe, pure, and potent. So let's turn our attention to the development of biosimilars in the U.S. And we'll start a little bit by describing the biosimilars program here at FDA. As of December 1st, 2017, 60 programs were enrolled in the Biosimilar Product Development, or BPD program. Most of the biosimilar activity around the development of these products has been within the Center for Drugs Evaluation and Research, or CDER, here at FDA. and we've received meeting requests to discuss the development of biosimilars for 27 different reference products to date. Since program inception, and as of December 1st, 2017, 11 companies have publicly announced the submission of 20 351K BLAs or marketing applications to FDA. And as of December 1st, 2017, 8 351K BLAs for biosimilar products have been approved, and I've listed the 8 that have been approved to date on this slide. FDA has also published guidance primarily for industry to provide advice and guidance as to the development of biosimilar and interchangeable products. The guidances listed in bold here are final, and the ones that are in regular font are still in draft. I've also provided a link at the bottom of this slide that will take you to a page where you can access all of these guidances and read more about the details that describe these guidances. But in the next few slides, I'll talk a little bit primarily about the scientific content of a couple of these key guidances. So as described in guidances, we describe the overview and the scientific thinking and some regulatory thoughts about FDA's approach to the development of biosimilars. And I'm going to go over a few key development concepts. The first key concept is that the goals of standalone and biosimilar development are different. So on the left-hand side, we have a green square, which is intended to depict a standalone or 351A development program. As mentioned previously, the goal of a standalone development program is to establish the safety and efficacy of a new product. You can see that the types of data that support a 351A application include analytical data, animal data, clinical pharmacology data, which can include phase 1 and phase 2 studies, which establish a dose that's carried forward, which are ultimately tested and evaluated in typically 2 large phase 3 efficacy and safety trials to support the approval of that product for a particular indication. In contrast, on the right-hand side, you'll see a blue triangle. And this is intended to depict an abbreviated development program, the 351K Biosimilar Development Program. Again, the goal of this development program is not to reestablish the safety and efficacy of the reference product. The goal is really to establish biosimilarity or interchangeability to a reference product. So how do you do that? You'll notice that the data types are similar to those of a 351A application. So it also includes analytical data, animal data, clinical pharmacology data, and what we term additional clinical studies. And I'll speak more to these types of data in the subsequent slides. However, you'll notice the different depiction as a triangle, and this is really intended to talk about the weight that we put on the types of data that are used to support approval. In contrast to a standalone development program where it's really culminating in the outcome of the phase 3 clinical trials, the bulk and focus of biosimilar development is flipped around, and we really focus on the analytical data to support that these products are highly similar. And then the clinical studies support the demonstration of no clinically meaningful differences. So the next key concept I want to cover is this idea of stepwise evidence development. FDA has outlined in guidance a stepwise approach to generate data in support of a demonstration of biosimilarity. And at each step of data generation, there's an evaluation of residual uncertainty about biosimilarity as that data builds. So you start with the analytical data as the foundation of a biosimilar development program, and after completing the analytical exercise, you stop and you say, what are the differences that I've seen between my product and the reference product? And that leads to uncertainty. So what is the proper test, or what can I do to evaluate the extent and potential impact of that difference? And that is where the subsequent pieces of a biosimilar development program come in. This concept of the data building on itself is what we refer to as totality of the evidence. And this is the approach in evaluating all of the data that supports biosimilarity. So there isn't a one-size-fits-all assessment. sponsors developing programs don't have the identical data. They all have differences that we look at specifically. And unlike a standalone development program where there are pivotal clinical trials that typically support the demonstration of safety and effectiveness in a particular indication, there's no one pivotal study that supports biosimilarity. We really look at the totality of the evidence of whether everything is pointing and trending in the right direction. The third key concept I want to talk about, and I've alluded to this earlier, is that analytical similarity data really is the foundation of a biosimilar development program. What the analytical similarity data includes is a large array of tests that really break down and look at the molecule at that structural and functional level. And it's a characterization of both the reference product and the biosimilar in a comparative fashion. And that really, as I mentioned, is the basis at that level of establishing that the products are biosimilar. So just a few words about proteins. Proteins are very large, complex molecules. These are very different from what we term small molecule drugs. These are difficult to characterize, difficult to manufacture. And so proteins have a hierarchy in terms of their structure, starting from the primary amino acid residue primary structure, moving on to alpha helices, and then subsequent tertiary and quaternary structures as the different amino acid chains fold and interact with each other. The heterogeneity of proteins is a result of the fact that these are products that are produced in a biological or living system, and there's an inherent variability with that. So these cells and living systems are sensitive to pH and temperature and the raw materials, and depending on the control of those factors, you can come up with different variants and different types of that particular product. So that is what is meant by lot-to-lot variability. Even with the manufacture of a single biological product, you'll see lot-to-lot variability because of this complicated nature of these proteins. All of these variabilities are carefully controlled to ensure that the variability does not impact safety and efficacy. And so all of these variabilities need to be evaluated in the analytical similarity exercise for the reference product and the biosimilar product. So in terms of generating the analytical similarity data, that's the foundation of a biosimilar development program, the applicant of the biosimilar product, the sponsor who's developing the 351K application and product, will characterize the reference product looking at its structure and its function. So from there, the reference product sponsor develops their manufacturing process, and in an ideal world, it's designed to produce a product with minimal or no difference in product quality characteristics compared to the reference product. We mentioned how complicated and large these protein products can be. And we talked about how there's inherent variability. And so because these are not small molecule generics, there will be differences identified between the proposed biosimilar and the reference product. And that in and of itself is not a problem. It's expected and normal. The key, though, is to identify what those observed differences are between the reference product and the proposed biosimilar, and to evaluate the potential impact of those differences observed. So what is the extent of the difference in terms of levels, and also what do we know from publicly available literature about what differences in that particular attribute mean. So if you really understand the relationship between a particular quality attribute and the impact of that attribute on the clinical safety and efficacy profile, this aids in the ability to determine uncertainty, residual uncertainty about biosimilarity, and to predict expected clinical similarity from the quality data. So I'll give you an example here. We know from extensive studies that certain glycosylation differences in a molecule can affect the pharmacokinetics, the PK, or exposure of the product in the bloodstream. And so if we see differences in the glycosylation profile of a specific glycan between the reference product and the biosimilar, the biosimilar applicant needs to evaluate the extent of that difference and show that the difference does not impact clinically. So in this case, if we have pre-existing publicly available information that can cause differences in PK, the appropriate test to do would be to do a PK study and to look to see whether the different PK parameters in terms of exposures, Cmax, for example, and AUC, we'll look carefully to see whether they differ. And if they do not, then we can see that even though there is a difference at the molecular level, that doesn't translate into something that has clinical impact. The fourth key concept I want to cover is the role of clinical studies in a biosimilar development program. We've talked about how the foundation begins with extensive analytical data, and then once an applicant has identified and evaluated differences that they see at the analytical level, you can start to think about what other studies are needed for a biosimilar development program. The nature and scope of clinical studies, and those are the clinical pharmacology and the additional clinical studies shown in the red box, the nature and scope of these studies will depend on the extent of residual uncertainty about the biosimilarity of the two products after we've done all the previous steps. And again, this gets at the stepwise evidence development that I talked about earlier. So in terms of clinical data, I'll describe the main categories and what we evaluate. As a scientific matter, FDA expects an adequate clinical pharmacokinetic, or PK, and pharmacodynamic, or PD, and that's only if it's relevant. We don't have PD markers for all of our products. We expect this comparison between the proposed biosimilar product and the reference product. In general, we consider PK and/or PD the most sensitive clinical study or assay in which to assess for differences between products, if these differences exist. And PD in particular is particularly powerful because PD is a measure of something along the mechanistic pathway. It reflects the mechanism of action or the biological effect of the drug. And if we can show at that level that the PD markers are similar, that's really important information that goes a long way towards showing that the products are biosimilar. Also, as a scientific matter, at least one clinical study that includes a comparison of the immunogenicity of the proposed biosimilar and the reference product will generally be expected. And this doesn't necessarily have to be a standalone study by itself. It can be incorporated into both the clinical PK and PD study that I just described, and then this next study, the comparative clinical study. So lastly, as a scientific matter again, a comparative clinical study will be necessary to support a demonstration of biosimilarity only if there are residual uncertainties about whether there are clinically meaningful differences between the proposed biosimilar and the reference product. And again, this is after we've done the structural and functional evaluation, the animal testing, the comparative human PK and PD study that I just described, and the clinical immunogenicity assessment. If we still need this information, then a comparative clinical study is appropriate. Again, there's no one pivotal study that demonstrates biosimilarity. It's the compilation of these clinical studies that go towards supporting the demonstration of clinically meaningful differences and ultimately for biosimilarity. So a few words about what I've called the comparative clinical study in terms of, it's probably most analogous to the phase 3 clinical trial that supports a standalone 361A development program. But we term it a comparative clinical study because it's a different development pathway, and the role of this clinical study is quite different. As I said, with a standalone, it's really pivotal and important to support the approval of a standalone product, the safety and effectiveness. Here, it's really the tip of that triangle that I showed you earlier. It's really intended to investigate at the last step whether there are any residual uncertainties and whether there's any question about the demonstration of clinically meaningful differences in safety and efficacy between the proposed biosimilar product and the reference product. The study population, the study endpoints, the sample size, and the study duration of a comparative clinical study should be adequately sensitive, again, to detect differences between products, should these differences exist. We're not reestablishing the safety and effectiveness of this product. The reference product, we've already established that for the reference product. Typically, for a biosimilar, we expect an equivalence design to be used for the comparative clinical study, although other designs may be justified depending on product-specific and program-specific considerations. And as I mentioned previously, there's always an assessment of immunogenicity that's built into the conduct of all clinical studies to support a biosimilar development program. So an adequate assessment of safety and immunogenicity is expected. The fifth key concept I'll cover is this, what we term extrapolation. So extrapolation is really the foundation of a biosimilar development program. It really gets at the heart, I should say, of an abbreviated licensure pathway. So the potential exists for a biosimilar product to be approved for one or more conditions of use or indications for which the reference product is licensed based on what we term extrapolation. Extrapolation is not automatic. An applicant has to provide sufficient scientific justification that extrapolation of their information from for one condition of use for other conditions of use is justified. We also note, and we've described the concept of extrapolation in our guidance, we note that differences between conditions of use, for example, indications, do not necessarily preclude extrapolation. So we have some products, for example, that are approved for rheumatologic indications as well as gastroenterologic. And just because those are different patient populations, that doesn't automatically mean that extrapolation isn't applicable, isn't warranted. It really depends on the scientific justification that a sponsor puts forward. And we've described in our guidance factors to consider and to address as part of the scientific justification that extrapolation is justified and warranted. And these include the mechanism of action in each condition of use, the PK and biodistribution in different patient populations, the immunogenicity in different patient populations, and differences in expected toxicities in each condition of use and patient population. So here I've tried to depict what we mean when we talk about extrapolation. So let's talk first about standalone drug development. So here we have the familiar green square. We're familiar with the data that supports the approval of a 351A standalone application. In particular, I'll draw your attention to the fact that there is generally a phase 3 trial or trials that supports the safety and effectiveness of each of the indications being sought by the applicant. So in the original application, they would have certain clinical studies that support approval, and if they wish to add additional indications to that product, they will need to conduct a separate clinical safety and efficacy trial for indication 2, 3, 4, and so forth. So now let's turn our attention to biosimilars and the familiar biosimilar pyramid. When we think about biosimilars, we talk about the demonstration that the product is highly similar with the analytical data and that there are no clinically meaningful differences with the clinical data. It's really this totality of the data, looking at all these elements, the analytical, animal, clinical pharmacology, and additional clinical studies data. It's all of these elements comparing the reference product to the biosimilar that feed into the concept of extrapolation. So we're talking about all of this available comparative data and information in the biosimilar application put forward by the biosimilar applicant. There's also the ability, as I mentioned, to rely on FDA's previous finding of safety and efficacy for other approved indications for the reference product. And this is scientifically justified because the applicant has been able to show that their product is biosimilar to the reference product. And lastly, the third sort of piece of information that supports the concept of extrapolation are those factors that I mentioned that are listed in our guidance, such as mechanism of action, PK, biodistribution, and so forth. It's the ability of the applicant to scientifically and appropriately justify all of those factors as to why it's relevant and applicable to their product. So it's the totality of these three pieces of information that biosimilar extrapolation is based on. And the last thing I want to point out is that extrapolation is not from the indication studied in the biosimilar development program to the other non-studied indications. It's not an indication to indication concept. It's really from product to product. So I'm going to switch gears a little bit and talk a bit about considerations in demonstrating interchangeability with a reference product. We do have draft guidance on this, and I'll speak a little bit about some of the key concepts that we've described in that guidance. Interchangeability is defined in the BPCI Act as the following. First, the biological product is biosimilar to the reference product. Next, it can be expected to produce the same clinical results as the reference product in any given patient. And lastly, for a product that is administered more than once to an individual, the risk in terms of safety or diminished efficacy of alternating or switching between use of the product and its reference product is not greater than the risk of using the reference product without such alternation or switch. The BPCI Act goes on to state that an interchangeable product may be substituted for the reference product without the intervention of the healthcare provider who prescribed the reference product. So FDA has published our interpretation of these statutory criteria in the draft guidance that I mentioned previously. And I want to mention that in the definition of interchangeability, you notice that being a biosimilar is one of the requirements. So for a biosimilar applicant who has come in first with an original application to have their product approved as a biosimilar, they may choose to come in later with what we call a supplement to later request approval as an interchangeable product. And if that is what they choose to do, that licensure as a biosimilar may be referenced to support a showing for the statutory criteria for demonstrating interchangeability, meaning they don't need to re-demonstrate biosimilarity if they're going to pursue interchangeability. That was the first step, and they can subsequently reference their own data that supported the demonstration of biosimilarity. Now, a biosimilar applicant doesn't have to seek all the conditions of use, for example, indications for which the reference product is licensed, but in their application for interchangeability, there is an expectation that sponsors or applicants will submit data and information to support a showing that the proposed interchangeable product can be expected to produce the same clinical result as the reference product in all of the reference product's licensed conditions of use or indications. Now, the data and information may vary depending on the nature of the proposed interchangeable product. And we do expect that the data and information that is submitted include a scientific justification as to why any differences that exist between the reference product and the proposed interchangeable product with respect to the factors described in the guidance do not preclude a showing that the proposed interchangeable product can be expected to produce the same clinical result as the reference product in any given patient. FDA does expect that applications for a product administered more than once to an individual generally will include data from what we've termed a switching study or studies in one or more appropriate conditions of use. Sponsors should evaluate the proposed product's presentation, including product design and user interface, relative to the reference product. And this is really to evaluate the impact of switching back and forth between the reference product and the proposed interchangeable product, and to see if that switching has impact. We have a number of factors that we've asked sponsors to look at in terms of additional data and information needed to support a demonstration of interchangeability. So I mentioned that the switching study is one data element that we ask sponsors to provide. And this is really intended to demonstrate that the risk in terms of safety or diminished efficacy of alternating or switching between use of the proposed interchangeable product and the reference product is not greater than if no such switch occurred. And this is really our interpretation of that description of the statutory language. So what does a switching study even look like? So the considerations for the design of a switching study will be product specific. It really depends on what you're trying to develop an interchangeable product to. And it should generally consider the scenario of switching, this alternating back and forth, where there is the most clinical concern for patients. So we sort of want to test the worst case scenario to see if there's any impact from switching back and forth. So for example, sponsors should consider the endpoints of a switching study. The primary endpoint should assess for the impact of switching on clinical PK and PD if available, because we believe that these endpoints are generally most likely to be sensitive to changes in immunogenicity or exposure that may arise as a result of alternating or switching. We generally think that clinical endpoints, long-term mortality, morbidity endpoints, are not sensitive to molecular differences between products. And the thinking is that with switching or alternating between products, what we may be concerned about is the development of antibodies and immunogenicity. And we think that PK and PD are the most sensitive and relevant markers to evaluate for the development of these antibodies and the impact of switching. As I mentioned, immunogenicity and safety should be descriptively analyzed. It's an important part of the evaluation, but these are generally evaluated as secondary endpoints. Applicants should also consider the study population. Is it adequately sensitive to allow for the detection of differences in PK and PD in the common adverse events that may occur and to assess for immunogenicity? So keep in mind whether the patient population is immunosuppressed, concomitant comorbidities, and so forth. Applicants should also keep in mind the condition of use or indications to be studied. These should be one for which the reference product is already licensed and should support extrapolation for other conditions of use. And lastly, the route of administration should study the route that will best assess how a patient's immune response will impact clinical performance. So for example, we know in general that the subcutaneous route of administration is more immunogenic than an intravenous or IV route of administration. And so an applicant, in terms of designing their switching study, should consider all of these factors before putting forward a proposal to FDA for consideration. A switching study should evaluate changes in treatment that result in 2 or more alternating exposures. So again, we term these switch intervals, but it's really getting at what is the impact of switching back and forth between the proposed interchangeable product and the reference product. And we've said in our guidance that at least 2 exposures to both products is needed. And lastly, sufficient scientific justification for extrapolation is necessary. It's the same considerations for extrapolation that we've described to support the demonstration of biosimilarity. So here's a schematic that gives you an example of what a general switching study design would look like. Again, we're looking at the impact of switching between the reference product and the proposed interchangeable product compared to just staying on the reference product alone. So let's start from the left and then move towards the right. So at the beginning, we expect an adequate lead-in period on the reference product. And then from there, there's a randomization to either what we call the reference product arm. So at the top, where patients who are randomized to that arm would remain on the reference product for the duration of the study. Another group of patients would be randomized to what we term the switching arm. And so this is the arm that would undergo a switch at the time of randomization to the proposed interchangeable product, would go back to the reference product, go back to the interchangeable product, and that's shown with the red color on this diagram. The primary endpoint is the intensive pharmacokinetic sampling that we described earlier, looking for the impact of the development of antibodies on PK and PD if relevant. We talked about the two exposures to both products, so you'll see that there's the reference product to the proposed interchangeable and back and forth 2 times. There's also, I should mention, pharmacokinetic cross-sampling. So in addition to looking at the impact of switching at the end of all of this, there's also sampling within the earlier time periods to get a sense of whether things are trending in the right direction. So after the intensive PK sampling, there is an adequate period of safety follow-up before the study comes to a conclusion. I want to talk a little bit about proposed presentations. So this is generally refers to whether the biosimilar interchangeable product is in a prefilled syringe or a vial. That's what we mean by presentation. So when we talk about interchangeability, a sponsor developing an interchangeable product generally should not seek licensure for a presentation for which the reference product is not licensed. Differences between the design of the container closure system or delivery device constituent part between the proposed interchangeable product and the reference product may be permissible and acceptable. But these differences really need to be analyzed appropriately. And this is what we've described in a subsection of that draft guidance for interchangeability as a threshold analysis. So what we're looking for are differences in product labeling, in the instructions for use, comparing the reference product and the interchangeable product. We're really looking for data to be provided to demonstrate that the differences between the presentations between the proposed interchangeable product and the reference product do not negatively impact the ability of the end users, and this includes both patients and their caregivers. It doesn't impact these end user groups to appropriately use these products when the interchangeable product is substituted for the reference product without the intervention of the prescribing healthcare provider or additional training before use. So as mentioned, the definition for an interchangeable product allows for substitution of an interchangeable product for a reference product at the pharmacy. And so we really want to ensure that a patient or their caregiver will be able to use an interchangeable product if there are slight differences in the presentation of that product. So we've talked quite a bit today, and I just want to summarize a little bit about what we've talked about. So again, the goal of a biosimilar development program is to establish biosimilarity between the proposed biosimilar product and the reference product. It's not to reestablish the safety and effectiveness of the proposed biosimilar product. It starts with stepwise evidence development with analytical studies as the foundation of a biosimilar development program. And it's really looking at the totality of the evidence, the data that supports the demonstration of highly similar and no clinically meaningful differences between products. FDA reviews all of this data and information, and if it supports the statutory standards for biosimilarity, then it's approved as a biosimilar. The product is then subject to the same types of post-marketing safety monitoring that any FDA-approved product would be subject to. So the approval of a biosimilar interchangeable product is based on an integration of various information, and it's really this totality of the evidence submitted by the applicant to provide an overall assessment that the proposed biosimilar is biosimilar to or interchangeable with the reference product. My final slide is just a brief word about using, from a practical perspective, biosimilar and interchangeable products. Currently, FDA has licensed 8 biosimilar products. Only a handful of those are actually currently marketed. But when it comes to using biosimilar interchangeable products, patients and their physicians can expect that there will be no clinically meaningful differences when taking a reference product and a biosimilar when these products are used as intended. All reference products and biosimilar products and interchangeable products meet FDA's rigorous standards for approval for the indications or medical conditions that are described in that product's specific labeling. Lastly, the FDA's high standard for approval of biosimilar interchangeable products means that patients and healthcare professionals can be confident of the safety and effectiveness of a biosimilar or interchangeable product, just as they would for the reference product. So with that, I thank you for your attention. If you are interested in this topic and would like to find more information, I've provided our website here that we've recently updated with additional information and education, especially for healthcare providers. And again, I thank you for your time.
Thank you, Dr. Lim, for sharing your expertise with us. Listed here again is the FDA's website for more information on biosimilars, as well as additional resources from the AGA and the Crohn's and Colitis Foundation. This concludes the webinar. Please return to gastro.org to complete additional activities.